Tolerance. Tolerance refers to an antigenic induced specific and responsiveness in tolerance refers to specific immunogenic non-reactivity to an antigen resulting from a previous exposure to the same antigen, while the most important form of tolerance is non-reactivity to self-antigen. It's possible to induce tolerance to non-self-antigen. When an antigen induces tolerance, it's termed tolerogen. Need for tissue and organ graft drove the curiosity to understand the mechanisms of tolerance. The observation a zygologist wins that dizygomatic bovine twins could accept graft from each other, but their sibling from other pregenesis could not tolerate such graft, led Medwar to perform a series of experiments to induce tolerance in mice. Here a zygotic twin in cow, and this is Medwar, a Nobel laureate. A series of experiments for induced tolerance. Strain B, strain A, the mouse, bone marrow transplant, strain A, newborn, after 8 weeks. He and his collaborator Brent and Burlingame, under the hypothesis that early exposure to foreign antigen might facilitate tolerance induction, performed this experiment. A white bee mouse made tolerant to C57 black mouse tissue. Tolerance to tissue and cell antigen can be induced by injection of hematopoietic stem cell in neonatal or severely immunocompromised by lethal irradiation or drug treatment animal. Also, grafting of thymus in early life result in tolerance to the donor type cells and tissue. Such animals are known as chimeras. These findings are of significant practical application in bone marrow grafting. Chimeric mouse. Factor affecting tolerance and the role of antigen. Factor which affect response. Tolerance to soluble antigen. A state of tolerance to a variety of T-dependent and T-independent antigen has been achieved in various experiment models. Based on this observation, it's clear that a number of factors determine whether an antigen will stimulate an immune response or tolerance. Here the table: factor which affect response, favor immune response, and favor tolerance. Physical form of antigen: the favor immune response, large aggregated complex molecule. Favor tolerance, soluble aggregate free simple small molecules. Antigen processing: favor immune response, properly processed. Favor tolerance, improperly processed. Route of injection: subcutaneous or intramuscular. Favor tolerance, oral or sometimes intravenous. Dose of antigen: optimal dose. Favor tolerance, very large or very small dose. Factor: age of responding animal. Favor immunosome, adult immunogenic mature. Favor tolerance, newborn mice immunogenically mature. Differentiation of state of cells: fully differentiated and memory, and differentiated B cell with only IgM T cell in the thymic cortex. Immunologic feature of tolerance: it's an antigen-induced active process. Like immunologic memory, it's antigen specific. Like immunologic memory, it can exist in B cells, T cell, or both. Like immunologic memory, it's easier to induce and last longer in T cell than in B cells. Induction of tolerance is very similar to induction of an immune response. Immunologic feature of tolerance: tolerance is different from non-specific immune suppression and immune deficiency. It's an active antigen-dependent process in response to the antigen. Like immune response, tolerance is specific, and like immunological memory, it can exist in T cells, B cell, or both. And like immunological memory, tolerance at the T cell level is longer lasting than tolerance at the B level. Mechanism of tolerance induction: clonal deletion. The exact mechanism of induction and maintenance of tolerance is not fully understood. Experiment data, however, point to several possibilities. Clonal deletion: functional immature cells of a clone encountering antigen undergo a programmed cell death. Autoreactive T cells are eliminated in thymus following interaction with self-antigen during their differentiation. Clonal deletion has been shown to occur as in periphery B cell expressing on IG, no ID, IgD on their surface when exposed to antigen are eliminated. Clonal deletion: thymus negative selection, bone marrow IgM positive, IgD negative B cells encountering self-antigen. Clonal energy: lack of co-stimulatory P7 molecule, exposure to large amount of antigen, improper antigen presentation. Here the macrophages. Clonal deletion: functional immature cell of clone encounter antigen undergo programmed cell death. For example, autoreactive T cells are eliminated in the thymus following interaction with self-antigen during the differentiation. Clonal deletion has been shown to occur also in periphery B cells expressing on IgM on their surface when exposed to antigen are eliminated. A negative selection of B cell in bone marrow. Clonal deletion: functionally immature cell of a clone encountering antigen undergo a programmed cell death. For example, autoreactive T cells are eliminated in the thymus following interaction with self-antigen during their differentiation. Negative selection. Clonal deletion has been shown to occur also in periphery B cell expressing only IgM, no IgD on their surface when exposed to antigen are eliminated. Clonal energy. Clonal energy: autoreactive T cell when exposed to antigenic peptides that don't possess co-stimulatory molecule B7-1, B7-2 become allergic to the antigen. Also, B cell when exposed to large amount of soluble antigen downregulate their surface IgM, become allergic and short-lived. These cells also upregulate Fas molecule on their surface, and interaction of these B cells with Fas ligand bearing cells result in the death via apoptosis. Receptor editing among B cells. Binding of B cell to self-antigen result in activation of RAG genes. RAG activation promote receptor editing and the change in specificity. Receptor editing: B cell which encounter large amount of soluble antigen as they do in the body and bind to this antigen with very low affinity become activated to re-express their RAG1, RAG2 gene. These genes cause them to undergo gene recombination and they change their specificity. Anti-idiotype antibody and anti-idiotype antibody produced permanently have been demonstrated to inhibit immune response to a specific antigen. Anti-idiotype antibodies are produced during the process of tolerization, and such antibodies have been demonstrated in tolerant animals. These antibodies prevent the receptor from combining with antigen. Suppressor cells: both low and high doses of antigen may induce suppressor T cells which can specifically suppress immune response of both B and T cells. Breakdown of tolerance. Termination of tolerance. Experimentally induced tolerance can be terminated by prolonged absence of exposure to the tolergen by treatment which severely damage the immune system, X-radiation, or by immunization with cross-reactive antigen. These observations are of significance in the conceptualization of autoimmune diseases. Termination. Clonal ignorance. T-cell reactive to self-antigen not represent in the thymus will mature and migrate to the periphery, but they may never encounter the appropriate antigen because it's sequestrated in inaccessible tissue. Such cell may die out of lack of stimulus. Autoreactive B cell that escape deletion may not find the antigen or the specific helper T cells, hence not be activated and die out.